All right, let's get straight into it. We're tackling the final 10 multiple choice questions of the A-Level Biology Paper 1. Now, this is that final stretch, right? It's where you can get tired, but honestly, nailing these questions is absolutely crucial for your grade. Okay, let's do this.
Look, these final questions are often where they test how well you can apply the big ideas when you're under pressure. So our goal today isn't just about spotting the right letter, it's about understanding the why behind the answer so well that you could turn around and teach it to someone else. Let's get these marks in the bag.
So, we're kicking things off with a huge topic, the circulatory system. The next 3 questions, 31 through 33, are all about how our blood does its amazing job, from hauling oxygen around to keeping our tissues happy.
Right, question 31. This is an absolute classic. The oxy-hemoglobin curve shifting to the right. The moment you see that phrase, your biology brain should be lighting up and screaming one key concept at you.
And that concept is, of course, the Bohr effect. So let's just quickly walk through it. You've got respiring tissues, like your muscles, pumping out loads of CO2. That CO2 jumps into the blood, reacts with water, and boom, you get carbonic acid, which then releases hydrogen ions. More hydrogen ions mean the blood pH drops, becoming more acidic. And here's the magic part, that change in pH actually changes the shape of the hemoglobin molecule. This makes it less sticky to oxygen. It basically forces the hemoglobin to let go of its oxygen right where it's needed most. And that whole process is described perfectly in option D.
Moving on to question 32, which is more of a clinical application. Now, the question gives us 2 massive clues. You have to spot them: initiated as normal and not correctly conducted to the ventricles. That tells us exactly where the problem is and where it isn't.
Okay, so let's trace the electrical signal's journey. Initiated as normal tells us the SAN, the sinoatrial node, the heart's pacemaker, is working perfectly. It's firing off signals like it should. The problem is the signal getting stuck on its way to the ventricles. So what's in the way? Well, after the SAN, the signal has to go through the atrioventricular node, the AVN, and then down through the Purkinje tissue. If there's a block somewhere in that part of the pathway, the ventricles won't get the message to contract properly. So the fault has to be with the AVN and the Purkinje tissue. And that makes A our answer.
And question 33 takes us right down into the tiny capillaries. We're constantly pushing fluid out of our blood to bathe our cells, but how on earth do we suck almost all of it back in at the other end?
So, it's basically a tug of war between 2 different pressures. At the start of the capillary bed, the hydrostatic pressure, the sheer force of the blood is high, and it shoves water out. But as the blood flows, that pressure drops. Meanwhile, you've got all these big plasma proteins that couldn't escape the capillary. They make the blood really concentrated, giving it a very low water potential. This creates a powerful osmotic pull, and by the end of the capillary, that pull is stronger than the push, so water floods back in by osmosis. That's our mechanism, which means D is the correct answer.
Okay, section 2. Let's switch gears and move up to the lungs. We're going to be looking at the structures that make breathing possible, from the big pipes like the trachea all the way down to the tiny air sacs.
Question 34. Seems straightforward, right? What do cilia do? But trust me, this is a classic trap. It's designed specifically to see if you're confusing 2 different but related structures.
And here's the absolute key distinction you need to remember. The goblet cells are the factories. They're the ones that produce the sticky mucus to trap all the gunk we breathe in. The cilia, on the other hand, are the workers. They are these tiny little hairs that beat in unison, like a conveyor belt, constantly sweeping that mucus upwards and out of our lungs. So cilia don't make the mucus, they move the mucus. That makes B the correct answer. C is the distractor they want you to fall for.
Now for question 35, we've got to compare and contrast. You have to think about what the trachea and the alveoli are built for. What are their jobs? And what tissues would they need to do those jobs effectively?
This table just lays it out perfectly. The trachea is your main airway. It needs to be able to protect itself with mucus, so it has goblet cells, and it needs to control airflow, so it has smooth muscle. But an alveolus? Its one and only job is super fast gas exchange. Its walls need to be incredibly thin, so adding layers of muscle or mucus cells would just get in the way. It has neither. Therefore, both tissues are in the trachea but not the alveolus, which makes A the correct answer. For question 36, we've got to interpret this image. We need to spot the cartilage. Now, whenever you see a cross-section of an airway like a bronchus, you're looking for these big, solid, almost glassy-looking blocks of tissue. You see where label B is pointing? That is a textbook chunk of hyaline cartilage.
Its entire job is to act like scaffolding to hold the airway open so it doesn't collapse every time you breathe. The other labels are just pointing to other layers. So B is our answer for sure.
All right, we are on the home stretch now, our final and biggest section, covering everything from nasty pathogens and how they spread to the incredible ways our immune system fights back. Let's do it.
Question 37. As soon as you read the phrase killed by penicillin, a huge neon sign should be flashing in your brain that says bacteria. Penicillin works by messing up the peptidoglycan cell walls of bacteria. So this question is really just a sneaky way of asking, which of these things do you find in a typical bacterial cell?
Let's just run down the checklist. Do bacteria have circular DNA? Yep. Do they have plasmids? Absolutely. What about 70S ribosomes? Yes, that's a key feature that makes them different from us. And a peptidoglycan cell wall? Well, yeah, that's the very thing penicillin attacks. So all 4 of these features are present in a typical bacterium. That means our answer has to be A.
Next up, question 38. Cholera. What is it about this particular disease that makes it so good at causing these huge, widespread outbreaks? We need to think about the pathogen itself and how it gets around.
This is basically the perfect storm for an epidemic. First, the main symptom, watery feces, means the infected person is shedding massive amounts of the bacteria into the environment. Second, it's spread through contaminated water, so you can infect an entire community really quickly. And third, in our modern world, someone can get on a plane and carry the disease to a different continent in a matter of hours. So are all 3 of these statements valid reasons? You bet they are. That makes A the correct answer.
Question 39, and we're diving deep into immunology. So your body has seen a pathogen before. You've got these incredible memory cells just lying in wait. When that same pathogen shows up again, the memory cells spring into action. But what do they actually do? What do they divide and turn into?
This comparison says it all. The first time you get sick, your immune response is kind of slow and weak. But the second time, it's a whole different ballgame. The secondary response is all about speed and overwhelming force. Those memory cells get activated and they start dividing like crazy, differentiating into a massive army of plasma cells. And plasma cells are your antibody factories. They just churn out tons of antibodies to neutralize the threat immediately. So, the division of memory cells produces a ton of plasma cells, which makes B our answer. And here we are, our final question, number 40. And it's one we can all relate to. Why on earth do we need a new flu shot every single year? The question tells us the virus mutates, but we need to explain the immunological consequence of that. This quote gets right to the heart of it. Your immune system works on recognition. Your memory cells are trained to recognize the specific shapes on the outside of the virus, the antigens. Think of it like a lock and key. But when the flu virus mutates, a process called antigenic drift, it changes the shape of the lock. So your old memory cells come along with the old key, and it just doesn't fit anymore. They don't recognize the virus, so your previous immunity is basically useless. You need a new vaccine to train a new set of memory cells for the new lock. That's why D is the correct answer. And just like that, we are done. We've powered through the final 10 questions, covering blood, the heart, the lungs, and the immune system. Let's just do a super quick recap of the absolute must-know points.
So, remember, the Bohr effect is all about CO2 and acid forcing hemoglobin to drop off oxygen. Heart block is a problem with the AVN or Purkinje tissue, not the pacemaker. In the lungs, structure is everything. Cilia are for moving, alveoli are for exchanging. And finally, your amazing immune memory can be fooled if a virus like the flu changes its disguise.
Hey, if this breakdown was useful for your revision, do me a favor and hit that like button and subscribe for more explainers just like this one. And honestly, for some of the best exam-focused biology content out there, you need to go and follow Mr. Chi on all his channels: Instagram, TikTok, Facebook, and YouTube. You won't regret it. And before you click away, I've got a question for you. What's the one topic that's really stressing you out for your exams? Let me know in the comments below. We actually read them, and your feedback helps us figure out what to break down next. Thanks so much for watching, and happy revising.